Dr. Carol Greider currently works at Johns Hopkins University as the Daniel Nathans Professor and Director in the Department of Molecular Biology and Genetics. Growing up, Greider suffered with dyslexia, and she has overcome many struggles throughout her education, such as her poor performance in English classes and standardized tests. She quickly grew a liking to science when she took biology in her senior year of high school because she realized that she did very well in classes that were more based on memorization, like biology and organic chemistry, in college, because her dyslexia did not interfere as much in those courses. Greider went to University of California, Santa Barbara, where she studied biology for her undergrad, and then she went on to UC Berkeley for her PhD in molecular biology. Greider has overcome many obstacles throughout her life. First of all, her mother passed away when she was just 7 years old. While growing up without a mom, Greider developed a sense of independence early on in her childhood. She also had difficulty getting accepted by schools due to her low GRE scores. She was actually only accepted by UC Berkeley and Caltech, which were the only schools that were willing to look past her test scores and struggles with dyslexia. Greider has 2 children, Gwendolyn and Charles, and she always prioritizes being a mother over her work. She also loves to read, cycle, and make friends and socialize with her fellow scientists in the lab. I admire Greider's passion for learning. Her strong will and her confidence has successfully gotten her through so many struggles throughout school and her postdoc research. She values collaboration in the lab, and she always says that research should be fun. I appreciate that her research is driven by curiosity. She never has an agenda and just follows where the data takes her. I identify with Greider on her journey towards gaining interest in biology and wanting to become a scientist, because I only discovered that I liked biology because I do better with memorization skills, and I always have a curiosity to discover more. Right now, Greider works in the Department of Molecular Biology and Genetics at Johns Hopkins University School of Medicine. Her research focuses on telomerase and how telomeres affect organisms when they are not functioning properly. She is working on finding how short telomeres cause DNA damage and which genes are involved, as well as the processing of telomeres and how elongation is regulated. The most recent paper that she has worked on and published is the Diagnostic Utility of Telomere Length Testing in a Hospital-Based Setting. This paper provides evidence that flowfish is beneficial for measuring telomere length in a hospital setting. This was done by studying blood cells of volunteers without a mutation, with a telomere mutation, and with bone marrow failure. The telomere lengths were measured for these groups using flowfish method, and by comparing the telomere length in lymphocytes of the different groups, the results show that the short telomere length is directly correlated to disease and aging. Cells treated with immunosuppression for bone marrow failure were observed to have longer telomeres than the cells of the original mutation. These findings are important in hospital settings because they can help doctors working on treatments for patients and diagnoses. Greider has discovered telomerase, which is the enzyme that elongates telomeres after cell division. She shares a Nobel Prize in Physiology along with doctors Elizabeth Blackburn and Jack Szostak for this discovery. In addition, she has found that harmful effects of too little telomerase lead to the shortening of telomeres, which causes degenerative disease and bone marrow failure. She has also found that too much telomerase causes tumor growth and a higher risk of cancer. She also studied genetic diseases caused by the inheritance of short telomeres. Currently, Greider is working with mice to study dyskeratosis congenita, which is a dominant telomerase mutation that causes death by bone marrow failure. In future research, Greider plans to determine genes that allow for DNA damage due to short telomeres and to discover more about how telomeres are processed and elongated.